Good afternoon, students. Today we're going to discuss about the structure and functional components of three other infectious agents. One is the viroid, second one is the prion, and third one is the mycoplasma. So start with the viroid. So now, what is viroid? Viroid basically acts as a plant invaders. That means it invade only in plants but not in animals. So the viroid is a virus like particle. Okay, so you can and I can give you an analogy like suppose some humanoid like robots, so where some robots acts and function like humans, so that is why that particular robots is known as humanoid robots. Just like that, in this case also the viroids acts and function like some kind of viruses. That is why the viroids are known as virus like particle, or vir, that means virus, and oid, that means the like. So viroid is a virus like particle. Now viroids are actually subviruses which is composed actually single circular strand of nucleic acid, that means the RNA, and it coat for a single protein. Okay, so viroid is basically composed of a circular RNA molecule without any protein coat. That means it is a totally naked single circular RNA molecule. Now, what are the characteristics of viroids? So, how it's differ from viruses? So, viruses, we have already known that virus is actually composed of either RNA or DNA. But in this case, the viroids is totally and wholly composed of a circular single-stranded RNA, and that particular viroid don't have any protein capsid or protein coat. That means the viroid is totally naked in form, and viroids are transmitted by aphids. Aphid means is a kind of insects. For example, some black flies, fireflies, these are all are aphids. Okay, and some plants that are affected by the pathogens, that particular viroid pathogens, are like potatoes, tomatoes, cucumber, and coconut, palms, etc. These plants are actually infected by that particular viroids. So viroids was first found by Theodore Otto Diner in the year 1971. It was first examined in the potato spindle tuber viroid that caused huge loss to the potato industry. So that Theo Diner in the year 1971, he actually discovered and isolated that particular viroids from a disease which caused the potato spindle tuber disease. Okay, and that particular disease is actually very infectious for the potato crops, and it losses that potato industry very hugely. Okay, so now the next point is viroids are actually plant parasite, and it hijack the transcriptional machinery of the organelles such as nucleus and chloroplast, and they are known to be non-coding. That means they do not code for any kind of proteins, rather they hijack the transcriptional and protein synthesis machinery of the chloroplast and infect that particular plant species, and they infect with the help of a phenomena which is known as the RNA silencing. Now, the only known disease in case of humans which is caused by that particular viroid is hepatitis D. Now, the characteristics. So, viroids are basically very low molecular weight RNA molecule, and obviously it is the smallest known agent causing any kind of infectious disease, and it reproduces within the host and cause variation in them and causing to the death of that particular plant. And viroids are formed only on the RNA and infect only plants. Okay, and viroids are moved through intracellular manner, cell to cell through plasmodesmata, and long distance through phloem. That means when the viroids actually transmitted from one cell to next cell or nearby cells or adjacent cells by the means of the one, so you can say one cell adhesion structure which is known as plasmodesmata, and in long distance through the phloem tissue. So you can see here, this is the structure of a viroid, and this is totally composed of a single stranded RNA molecule and the circular single stranded RNA with some pairing. There are some pairing is also occurred between the complementary base sequences found in the single stranded RNA, and where the complementarity is missing, loops are formed. These are the loops where the complementary is missing. Okay, now go to the next infectious agent, which is known as the prion or prion. Okay, now the characteristics or some introduction, that the prion is actually pronounced as prion, and it is a shortened term for proteinaceous infectious particle. Okay, and it causes a particular disease which is known as transmissible spongiform disease, which attacks the central nervous system or in the brain. Okay, now the differences from bacteria and viruses, that prion do not contain any kind of nucleic acid, rather they have a protein structure and they don't have any RNA or DNA like things. Okay, and it is very sensitive to radiation, for example some alpha, beta or gamma radiation, and they are extremely resistant to heat and chemicals, and they are not get destroyed by any enzymes which can degrade DNA or RNA. For your knowledge, I can share one information that DNA can be degraded by some DNase enzymes, and the RNA which also can be degraded by some RNase enzymes. So these two enzymes are responsible for the degradation of DNA and RNAs, but this particular prion protein is totally resistant for this kind of enzymes. Okay, so no enzymes can destroy or can degrade that particular prion proteins. And now this last point, the prions are very difficult to decompose biologically, and they can survive in the soil for many years. So they are totally undegradable proteinaceous substance. Okay, now the basic structure. The basic structure of prion is made up of the normal prion contain about 200 to 250 amino acids, which is twisted to form a telephone wire-like structure. These are the telephone wire-like structures, which is also a alpha helical structure, and some tails. The tails means these are the tails. These are where the no helical structure is formed. So if you ask someone that how the prions or how many amino acids are there in prion, the answer will be there are 200 to 250 amino acids which is twisted in a telephone coil or telephone wire like fashion to form a alpha helical structure, and that particular alpha helices are joined with each other by non-helical structure and form two different tail structures, two different tails, which is composed of some more amino acids. Okay, now how the mutated form looks like. So you have already know that the normal protein is made up of about 200 to 250 amino acids, and the mutated form or infectious form built with the same amino acid, but it looks a bit different. Okay, you can see here that in normal form there are about 3 alpha helical structures are present, and 1 or 2 beta sheets are present, and these are the linking portions or where the non-helical forms. But in case of the mutated form, the mutated form of the prion is made up of exactly the same number of amino acids or with the same sequence, but the structure being different. You can see here that there are about 2 alpha helical structures and 3 or 4 sheets are present. Okay, and these prions are basically about 100 times smaller than the smallest known virus. Now the nomenclature of the prion protein. So the normal prion protein is also called the PRPC alpha helices, and the mutated form or the abnormal form is known as PRPSC for scrappy. So what is this scrappy? I will come later on. So this is the mutated form which is called or which is known as PRPSC, and which is also known as the scrappy protein. Now the most important part of this particular prion protein is that abnormal form or PRPSC, that form has the ability to convert the normal form into itself. Okay, so this is the most important criteria of prion proteins. That means the abnormal form can convert the normal form into itself. Okay, now the causes of prion disease, that PRPC or PRP-sen is produced by normal or healthy cells. The sen in this case means the sensitive protein, because that particular protein is totally sensitive to being broken down by some kind of enzymes. But the PRPC is present mainly in neurons and some other kind of cells. So the cells, the prion proteins, the normal prion proteins which are involved in the which is present in the neurons, are basically involved in the communication between the neurons, cell death, and sometimes the sleep pattern control. Okay, so this normal protein is associated with several important functions which is occurred continuously within the neurons. But the second type, or the abnormal form, that means the PRPSC or PRP resistant, in here the res means the resistant, because it is very much resistant to being broken down by some enzymes, and this particular abnormal protein is responsible for a particular disease which is known as the spongiform disease, which is found in the brain, and this form is very much resistant to being broken down. That is why this is known as the PRP resistant or PRP abnormal or PRP scrappy protein. Okay, now the particular prion do not have that any kind of genetic material, either RNA or DNA, but the question comes that how, once they get infect some normal individual or normal cells, how they can replicate. So how this is possible? So several experimental evidence support the idea that when PRPC comes into contact with PRPSC, it is converted into PRPSC. So the result is a chain reaction that multiplies the copy after copy to the infectious protein or infectious prion. So you can see here that the blue one is the normal protein. So after some kind of misfolding, the normal prion is converted into a mutated form or abnormal or scrappy form. So now when the mutated form is there and some normal protein is being attached with the mutated form, again after some time some autocatalytic conversion is occurred and the normal form is converted into a mutated form, and this cycle goes on like this. You can see here there are about 3 mutated forms present. So after some time, the prion replication, the aggregates fragment to form new seed. It is a seed, so it again function like this, and that particular seed is again comes in contact with a normal prion protein or PRP sensitive protein, and again the cycle begins like this. Okay, so this is a process by which the normal prion get converted into a mutated form, and when the mutated form is produced, the mutated form replicate itself in a autocatalytic conversion. Okay, so this is how the mutated form of the prion or PRPSC protein is replicating itself by a autocatalytic conversion. Okay, and finally this abnormal shape, when the abnormal shape is produced, that means that when the PRPSC protein is produced, it tend to stick with each other, and over time that abnormal protein stack up to form a long amyloid fiber structures, and these amyloid fibers are very much toxic to the cells and ultimately kill the normal cells or normal neurons. And after some time, the astrocytes, which is a neuronal macrophages which crawl to the brain where the amyloid fibers are present and the dead neurons are present, and finally it engulfs the dead neurons, and the amyloid fiber still remains, and this process makes the brain a sponge like structure or spongy form. And this is how the prion protein is responsible for that particular spongiform disease. Now, history, a little bit of history about the prion disease. Now, the scrappy. Now, how the scrappy term comes from? So, in 1730, an earliest written record was found regarding the scrappy disease is found in an English sheep, which is prevalent condition found or prevalent disease found in Central Europe. So this particular disease is responsible for continuous or intense itching of the sheep, and finally the sheep scrap off their wool, and this is how that particular scrappy form or scrappy term comes for this to describe that abnormal prion protein. Okay. Now that particular disease which is known as the Creutzfeldt-Jakob disease or CJD. So this Creutzfeldt-Jakob disease is a same kind of progressive mental or motor neurological deficit disease found in young human patients. So this is also a neurodegenerative central nervous system disease which is characterized by some kind of seizure, massive incoordination and dementia, and the occurrence is about 1 in 10 lakh. So this particular disease is occurred only in humans. Okay. Now another example comes from a particular disease which is very much ethnic in 4 tribe of Papua New Guinea. That particular disease is known as the Kuru, and this particular disease the recognition of that particular disease comes in the year 1950. So this Kuru disease is also a transmissible disease and occurred during a very strange ritual funeral process in which the brain of the dead tribe members was removed from the skull, cooked and finally being eaten. So it is a very strange kind of a ritual which is found in that particular 4 tribe of Papua New Guinea, and finally that cannibalistic behavior finally lead to the mortuary of that particular tribe, and the disease is extinguished from that particular tribe finally. Okay. Now that this is another disease which is mostly found in bovines or some cattle, which is known as the mad cow disease. And this mad cow disease is very much strange, where the animals display several kind of aggression or some abnormal behavior like nervousness, abnormal posture, very much temperament and aggression. You can see that particular cattle show very strange behavior. So finally that several prion disease are found in humans and animals. In humans, that Creutzfeldt-Jakob disease, that fatal familial insomnia, sporadic insomnia, Kuru, and GSS, that means Gertzmann-Straussler-Schreckner disease. So this, and finally in animals, in scrappy, in goats and sheep, that BAC or mad cow disease, which is also known as the bovine spongiform encephalopathy. And finally the chronic wasting disease, which is found in deer and elk. All are an aggregative form of a particular disease which is known as the transmissible spongiform encephalopathy, okay, which is caused by that particular prion protein. So he is the man, Dr. Stanley Prusner. He gave a hypothesis which is known as the Prion hypothesis, or proteinaceous infectious particle, or Prion hypothesis. So this hypothesis states that the PRPSC, which is the abnormal protein, which is very infectious, and the highly purified form, that particular PRPC protein, is very much infectious, and also that prion is also replicating protein. That means it can replicate itself. Okay, and for this discovery or hypothesis, he won the Nobel Prize in Medicine in 1997. Okay, now there is a slogan regarding that particular prion infection, that there is no cure for mad cow disease or Creutzfeldt-Jakob disease. So the prevention is the only available option to get you can say that to get uninfected from that particular disease. Okay, now finally the mycoplasma. So now the mycoplasma, what is mycoplasma? So basically in 1896, two scientists, Knockert and Rocks, reported the cultivation of a causative agent which caused a contagious bovine pleuropneumonia in cattle herds. So finally the two scientists represented the first isolation of PPLO, that pleuropneumonia like organisms. Okay, and finally the name mycoplasma is given for that particular pleuropneumonia like organisms, which refer to organisms similar to the causative agent, or CBPP. Okay, so the mycoplasma comes from two Greek words, that mycos means the fungus and plasma means the formed. So mycoplasma is basically a fungus like organism which cause that particular pleuropneumonia disease in cattle. Okay, now the mycoplasma are basically the smallest living organism ever observed, and these organisms are unique among prokaryotes, that they lack any kind of cell wall. Now the classification, the family Mycoplasmataceae is the family where that particular mycoplasma is categorized, and that they require cholesterol and other sterols as essential growth factor for their reproduction and survival. So basically two genus comes under this Mycoplasmataceae family. One is the Mycoplasma, which require or utilize the glucose and arginine for their growth, and they do not split urea, whereas another genus which is ureaplasma, which hydrolyze the urea, and they do not contain, they do not utilize that particular glucose or arginine for their growth. Now the basic characteristics of mycoplasma, they are basically unicellular prokaryote. The size varies from or ranges from 150 to 250 nanometer. The genome size is very small. It is about 800 kilo base pair or kbp. No cell wall is there. That means they are totally resistant to penicillin, cephalosporins, and vancomycin and etc. such antibiotics, and they contain a sterol-like cell membrane. They show a fried egg or mulberry colonies on agar. Agar means it's a culturing media where several bacteria are cultured for their growth and reproduction and for experimentation. They show some kind of facultative anaerobes, but the exception is there, that pneumonia, which is strictly aerobic in nature. They grow very slowly by binary fission. The doubling time can be as long as 16 hours and extended the incubation needed. So the doubling time means they get doubled, the number get doubled after every 16 hours, and the extended incubation is needed, that extended incubation period is needed for their doubling. Okay, they require a complex culture media for their growth, including the sterols, And major antigenic determinants are glycolipids and proteins. Here the antigenic determinants means which functions as an infectious molecule to infect the host cell. That means they are antigenic in nature, or they act as an antigen, and the antigenic particles found in the plasma membrane or of the particular mycoplasma is basically chemically glycolipid and proteinaceous in nature. And one can differentiate different types of species according to their requirement of growth factors. For example, the Mycoplasma pneumoniae, which require glucose as their growth factor, Mycoplasma hominis, which require arginine, Mycoplasma ureolicum, which require urea, but the Mycoplasma genitalium is very much difficult to culture. Now the colony, how the colony looks like. So Mycoplasma basically cultured in the agar medium for several days and with a long incubation period. They can be visualized under 30 to 100 magnification microscope, and the central portion of the colony penetrates the agar. You can see here, this is the central portion. Okay, so central portion can be penetrate on the agar, whereas the periphery is spread over to the adjacent surface, and this structure is quite similar in observation, or it observed quite similar, or it looks like similar like fried egg in appearance. Okay. So why Mycoplasma is pathogenic in nature? They don't have any kind of cell wall, and this particular characteristics probably facilitate to make a close contact with the host cell and to exchange the compound, the toxic compound between the bacterium and the host cell, and as a consequence of this bacterial surface parasitism, the host cell is severely damaged. And finally, the exchange of toxic metabolic compound is discussed as possible cause of the cell damage. So the toxic agents which is found in the bacterium is get transferred from the bacteria to the host cell, and ultimately the toxic substance make a possible damage to the host cell. So this is up to your first part. So within today or tomorrow I'll give you the assignments, some questions, and you have to answer the questions and submit it to me as soon as possible. So stay safe, stay home and study hard, and thank you for your attention.